Welcome to Face Forward, the podcast, the series that takes you behind the scenes of the riveting world of clinical trial management. I am your host, Valerie Coveney, and I am thrilled to be your guide on this journey through the nuances of planning, executing, and navigating the clinical research landscape. Joining us today is the head of the early phase department with more than 20 years experience in translational and clinical research. We will dive into study design considerations of early phase trials in dermatology. Let's extend our warmest welcome to Innovaderm's head of the early phase department, Ana Palijan. Thank you for being here.
Thank you so much for having me.
So I would like to say before we get into this episode is that Ana and I work together and collaborate on different projects. And one thing that I can say with all honesty is that Ana always gets back to data quality. Am I right?
Oh, yes. You know how they always say in real estate, location, location, location? Well, I say quality, quality, quality.
Exactly. So, Ana, before we go any further, what would you say makes early phase trials unique or different from later phase trials?
The real difference, what I find about early phase trials versus later phase trials is this is the beginning of a new story in the drug development. We are out of the preclinical. We have an asset that actually has a potential of going on the market. So this is a very unique and also a serious, responsibility that we have here. Of course, when we think about early phase, you have the SAD, MAD. And usually, depending also the type of IP that we have, this is a systemic, you will go into the healthy population. And then you move to the patient population, and usually you will have those in the 1B or 2A. And I'll be honest with you, 1B and 2As, this is what I really love, is when we get into the patient population. And this is where the study design is different because now we're looking at the safety. Usually that's your primary endpoints. We want to make sure that what is the level of absorption. And also it's our first hint at seeing if the drug works. We can have actually looking at the efficacy of the drug. And dermatology, which is even more interesting, is you see it. You see the lesions on your skin. And when we focus here, let's say on an inflammatory disease, right? And you can really see how the disease progresses with treatment. And how do we measure this? Dermatology is complex. It's different. It's actually PIs with visual symptoms will evaluate visually how these are clearing with time, if they're clearing with time. So this is where I think it's early phase is really everyone. Sponsor sites, CRO, researchers, we really have an impact on how we're going to write this story. And it's all about maximizing the value of your program, but also is being strategic in our decisions.
And if we circle back here to data quality that we mentioned earlier here, how would it come into play for the early phase?
It's all about data quality control early phase because, like I said, this is where we're starting to write the story. These are the first chapters of the book. So the first words that you write down are critical. And also investment is the part of the sponsor, right? This is budget. This is finances of the study of the trial itself, the funding. So we have to be really strategic. This is usually where we're going to have a few patients, a smaller population, where we're going to be doing these safety profiles, looking a little bit at the efficacy if there's any hint of it, these proof of concept studies. So every data point is critical. Let's say I'm doing a study in atopic dermatitis and I'm treating them, let's say for 4 weeks. I want to see a change from baseline after treatment as one of the endpoints to start to see like in a proof of concept study. I cannot have a strong placebo responder rate here. If I have only 40 or 50 subjects, like in a 1-to-1 or 2-to-1 randomization of, let's say, placebo versus the IP, the treatment, I cannot afford to have a high placebo responder rate because I'm running to have a false negative study. Because are the subject actually responding or not? I don't know. And how can I have that in dermatology? Well, this can happen actually because either how the protocol was written would allow other treatments. This is skin. Your skin is dry, it's itching, it's burning. You can put emollients, you can put creams. Maybe that's affecting my lesion. Or I can have inconsistencies in how the severity of the lesion is assessed. Usually we have to go multi-centers. We have to do many sites in a study, in a trial. So this is where every PI has to be really well trained to assess the person himself or herself themselves consistently. And also it has to be done consistently throughout the sites. If not, we will run into this signal problem. So yes, early phase studies, trials, it's quality, it's all there from day one to the last day. Yeah.
Exactly. So I gotta say, Anna, one thing we really do have in common is the excitement about what we do. And I just see how excited and how passionate you are about your projects. And it really reminds me a few months ago when we worked on this article for the website. It was on, I think it was skin microbiome.
Yes, good days.
Yeah. And we had the exact same type of exchange as today. And it kind of brings me to think, how does these types of derm techniques integrate into the design of the early phase trials?
Yes, so I'm really going to focus within the phase 1B or 2A proof of concept, really when you have access to the patient population, right? And absolutely, these techniques. This is my favorite part. I'll be honest with you. And then this is also, and when we are designing the trial, the program, and also sitting down with the sponsors and saying, hey, what can we do to get more data, more information on your IP? And this is it. These exploratory endpoints. It really brings value to the program, to the asset. Not only can we show, oh, look at this molecular signal, but also it can be very strategic for the sponsor to know, is there any crosstalk? Is there any other things signaling that's happening? And sometimes with this data, you can actually go back to the bench and design another drug that can do something similar in that pathway. You can actually get really important information.
Yeah.
Or even more importantly, you can even see, oh, this pathway is affected. Therefore, it can actually be interesting to look at this other indication. So, yes, it brings absolutely a lot of value. And yeah, there's so many fun things that we can do, right? Because it's dermatology, where skin is right there. We can touch it. It's easy access to the specimens. We can do genomics, all the omics, right? We can do gene expression. We can do skin biopsies. It can be done by tape stripping. It can be done so many things. And then skin microbiome. This is something really exciting that a lot of more publications are out there. And even if I just take a step back, I really find that in the last 4 to 5 years easily, translational research in this field has really exploded, and we really see how the pieces are coming together now, how we can relate the omics that say, oh, I have this lesion severity, and when I look at the skin microbiome, I can say, oh, well, I have this different type of presence of population of the biota or shift the biota. And this is really like doing this correlation of what we see as a severity and then actually what's happening in the molecular world or the omics world. Same thing with lipidomics. And also, how can we design this in a way where I can get information or tissue material that I need to answer these questions, but still make it feasible for the sites to perform these experiments, these collections, and also make the experience fun for the subject. Because patients who participate in studies, this is actually even more so for early phase trials, this is for research reasons. They're really giving their time for research to really understand, to have more information. And this is where these exploratory endpoints actually maximize, their input in science.
Exactly.
So absolutely. I find in early phase, this is the time where we can ask all these fun questions, get all these fun answers. And then from there, can actually shift maybe the focus of the program. Like I said, bring a lot of value to the program. And also because we're working in a smaller population study, it's more manageable to do all these type of sample collections. But this is also where quality is critical. As before, we're dealing with a small number of population with multi-sites type of study designs where many different sites across countries are doing these collections. So what do we do to make sure that everyone's doing the same thing the same way, storing it the same way, that the sample's integrity remains while they're shipping to the labs when we're doing the analyses? It's always there. And what I think it's really fun is how can we put the pieces together? Like, how can we make this operationally feasible? It's like a big puzzle. That's what I like.
Yeah.
And when you're writing a story, it's like how everything fits together.
Exactly.
Yeah.
Great. So, Anna, I feel like we could continue for hours, but this has to come to the end of the episode for today. So I just want to thank you so much for being here and taking the time to answer our questions for our listeners today.
Thank you so much. And I hope you're going to call me up for other subjects. I really love talking with you.
We will. I promise we will. As we conclude another illuminating episode of Face Forward, we find ourselves at the crossroads of science and progress. Remember that behind the jargon and statistics lie stories of unwavering commitment, meticulous observation, and the pursuit of evidence that shapes our understanding of health and disease. Stay at the forefront of knowledge and innovation and follow Face Forward on your preferred platform. My name is Valerie Kovney. Thank you for joining us. Until next time.